Hello. Archaeologists count things all the time. In comparing sites or layers within a single site, for example, we often want to know if some kinds of artifacts or plant or animal remains are more abundant than in some other site or layer. We also make use of quantification that's not precisely about counting. For example, we might want to know how often at least one of something occurs, a particular species of legume perhaps, in a number of storage pits or 10-liter sediment volumes. Or we might want to quantify how diverse some assemblage is. That doesn't just involve counting specimens, but also the number of kinds of things to which those specimens belong. In today's video, I'll just discuss some of the ways that archaeologists have tried to quantify things. As you'll see, some of them are problematic, if only because of the impacts of sampling and poor preservation. But it's important to keep in mind the goals of quantification if you're to select a measure that is most effective for your particular purposes. A very common type of archaeological analysis involves comparing assemblages or sites or other kinds of contexts with respect to the proportions or percentages of various categories of items, such as animals. However, that presupposes that we have some reasonable means for quantifying the items that go into each category in this comparison. The simplest kind of quantification in archaeology is a measure of abundance that just involves counting things. In fact, some people say that one of the things that archaeologists do the most is count stuff. So remember the video I did previously on classification. One of the reasons we make classifications is it allows us to compare and contrast sites and deposits and so on the basis of the relative abundances of different categories of material. So here I have some pottery sherds that I've classified into 6 categories, and this most simple kind of quantification is simply a count of fragments. Its archaeological name is NISP, which stands for Number of Identifiable Specimens. So it means we just count any fragment or whole piece, as the case may be, that can be identifiable to a particular category. There may be some potsherds or whatever that we can't identify, so they don't get included in the count. So here we have, category 1, whatever you might call it, which is kind of pinkish wheel-made pottery. We just count them. 1, 2, 3, 4, 5, 6, 7. So we have 7 of those. We record in our database that we have 7 of those ones. These nicer painted ones, 1, 2, 3, 4 of those. And we record that. These rather crude vegetable tempered sherds. We have 3. These black slipped sherds with burnish, I think. We have 4 of those. These wheel-made sherds with a pale slip over a red body, we have 4 of those. And these pinker combed potsherds, 1, 2, 3, 4, 5, 1, 2, 3, 4, 5, 6 of those. That's it. So we just count the fragments, and we don't worry about the relationship of the fragments to whole pots. Just the ratios of these sherds to one another will tell us something useful, but they're not going to tell us the ratio of different kinds of pots to each other, because this kind of pot might break into more pieces on average than, say, this kind of pot or this kind of pot. A number of factors contribute to the value of NISP. One is the number of individuals, in this case pots, in the population. Another is fragmentation rate, which is just the average number of pieces into which an individual breaks. A third one is recovery probability, which is really a combination of several factors, including the probability of being deposited where the excavation took place, which depends in part on the use life of a vessel, the probability that some archaeologists would recover it in the excavation, and the probability that it would be correctly identified. In the case of some kinds of individuals, notably animals, it's also influenced by the number of distinct elements in that individual, such as bones. For pottery, that last factor would normally be equal to 1. Thus, you'll sometimes see the expression NISP equals NRSF. This is not intended as a way to calculate NISP. After all, to calculate NISP, you just have to count things, but rather as a way to express the various factors that contribute to that value. When we're able to reconstruct a vessel, as you see in this case, we can get some idea of how many sherds a vessel of this type and size would likely break into. So in this case, it broke into including the sherds that appear to be missing, it broke into a little over 20 sherds. So that gives us some idea of the fragmentation rate for this particular kind of vessel. Of course, it's only based on a sample size of 1, so we would expect the actual range of breakage to vary a little bit around that figure. And we would also expect that a larger vessel of the same type might well break into more sherds than that, and a smaller vessel would break into fewer sherds. Furthermore, if you had a vessel that was made out of a stronger fabric or that had thicker vessel walls, it might break into fewer pieces as well. So there are a number of factors that affect the fragmentation rate in vessels, all of which will affect, of course, the value of NISP, the basic count of sherds. The problems of quantification provide another example where probability comes into play. As noted in my video on probability in archaeology, we can compound the probabilities of outcomes that are not mutually exclusive to get a sense of how commonly some combination of events might occur. For example, it could be that, on average, thick-walled pots break into fewer pieces than thin-walled ones do, or large pots into more pieces than small pots. It could also be the case that some kinds of pot have short use lives, meaning that they only typically last a few weeks before they get broken, while other kinds of pot that people use rarely or curate very carefully might last for many decades. All these would have an impact on the probability of encountering sherds from those pots in an archaeological deposit. We can model all of those factors with probabilities based perhaps on experiments or ethnographic observations to see how they could affect the relative abundances of sherds in archaeological deposits. While it's easy to account for some aspects of recovery probability, such as the percentage of a site or a deposit that the archaeologist includes in a sample, differential use life can be a particularly thorny issue. In the example here, with 4 different kinds of pots one might find in a kitchen, each type of pot could differ markedly in the number of months or years it is used before it is discarded or broken. If we actually knew these use lives, we could calculate how many would be discarded over a period of, say, 5 years. Such differences in use life can have a huge impact on the number of vessels that actually enter the archaeological record, a problem that differential fragmentation will only exacerbate. However, these problems are not unique to NISP. Minimum number of individuals, or MNI, is one of the simpler methods that archaeologists have used in order to get some idea of the number of animals that contributed to an assemblage of animal bones, or its parallel, minimum number of vessels, has been similarly used to try to get an idea of the number of pottery vessels that contributed to an assemblage of pottery fragments or sherds. Here I'm going to try to demonstrate that in a very simple way with just a few elements of a caribou skeleton, or several caribou skeletons actually. And ordinarily we'd be looking at a lot more than 2 kinds of elements, but here we're going to just look at tibiae and humeri of the caribou, just to demonstrate how MNI would work. So here we have the tibiae of the caribou. We have 3 left tibiae here, and we have 3 fairly complete right tibiae, as well as 1 fragment of the distal end of a caribou tibia, right tibia. Now that tells us, using this assemblage here, that at least 4 caribou must have contributed to the assemblage, because we have 4 right tibiae, and each caribou has 1 tibia, or 1 right tibia and 1 left tibia, so we use whatever's the maximum number of elements in order to get our number of animals. So if tibiae were the most frequent element in the assemblage, we would end up with an MNI of 4, using the simplest form of MNI. Similarly, we can look at the humeri here. In this case, we have only 2 right humeri, but we have 4 fairly complete left humeri and 1 distal half, almost a half of the distal end of the humerus, the left humerus. So this tells us, using the simplest method of MNI, that there must have been at least 5 caribou that contributed to the assemblage, because we have 5 reasonably complete left humeri. And since that exceeds the number that we have from the tibiae, we would now conclude that the MNI value should be 5. Again, if we had some other element that was more numerous and had either a left or a right element that was more numerous than 5, then that would elevate the MNI to whatever number that was. This version of MNI, then, is just the number of the most abundant element. For example, if 6 left humeri and 2 distal ends of left humeri were the most abundant elements, we would have an MNI of 8. However, some analysts use a more complex version of MNI. This can include comparing fragmentary elements to see if 2 fragments could have come from the same bone. This involves checking for overlap between 2 fragments. In this case, they do overlap, so this would count as 2 elements and not 1 when we calculate MNI. Analysts who use this more time-consuming method obtain higher MNIs than those who use the simple method. Another elaboration of MNI involves comparing left and right elements to see if they could have come from the same animal. Just because we have 5 left humeri in this particular example, doesn't necessarily mean these 2 right humeri came from 2 of the same animals that are represented by the left humeri. In fact, statistically speaking, that would seem pretty unlikely. Assuming these were just a random sample of the animal bones in an archaeological site, and in a previous video we've talked about, or I've talked about sampling issues, it's pretty unlikely that a typical cluster sample from an archaeological site would have all of the lefts and all of the rights match up with each other. Where they show strong symmetry and almost identical size, the assumption is that they come from the same animal. So the value of MNI is no different than the simple method. If, by contrast, they differ quite a bit, we would assume they come from different animals, and so additional bones can be added to the count for MNI. So one of the things some archaeologists do is they proceed with a matching procedure. They take each of the left elements that we have here, in this case 5 of them, establishing our MNI of 5, and we try to match them with the right elements to see if any of them could have come from the same animal. And here, for example, you can see quite clearly that they don't come from the same animal because they're radically different in size. And in fact, those can't match either. And we do this in turn with all of the lefts matching with both of the rights. And we don't see good matches. That's also not a good match. That's not a good match. Now this one might be a match. I'm not an expert on this because I'm not a zooarchaeologist, but let's say for the argument that this pair is a matching pair. I think it's probably not, but it's fairly close, so let's say these 2 match, which means these 2 probably come from the same animal, or at least could come from the same animal. So that means that we can ignore this one because it's already included in our MNI, but this one, this particular right humerus, doesn't match with any of the left humeri we have. It's clearly much bigger than most of them. In fact, I think it's bigger than all of them. So this means that there must have been at least one other animal, which means our MNI should be 6 and not 5. At least for the more abundant kinds of faunal remains, this method tends to lead to higher values of MNI than would result from the simple method. Both of these elaborations on MNI are more realistic in that they recognize that just because 2 different elements could have come from the same animal doesn't mean that they do come from the same animal. To summarize, the simplest form of MNI is nothing more than the number of the most abundant element. More time-consuming versions involve checking for overlap between fragments of elements and matching left and right elements to see if they could have come from the same animal. In any case, whichever form of MNI you use, it's important to let other researchers know which kind of MNI you are using. However, all versions of MNI underrepresent the most abundant taxa because they treat all bones and bone fragments that are not demonstrably from different animals as though they came from the same animal, even though that is usually statistically unlikely. A taxon represented by a single bone or bone fragment will get rounded up to an MNI of 1. Meanwhile, a taxon represented by hundreds of bones that probably came from dozens of animals might get an MNI of only 1 or 2 or 3. The typically uneven representation of different elements from the skeleton proves that many elements are missing from the sample. Not only does this mean that we should not expect every animal that contributed to the sample to be represented by the most abundant element, it also means that some animals probably aren't represented by any elements at all. For that reason, MNI tends to work best for catastrophic deposits, such as the remains of a feast or a massacre, in which most skeletal parts are represented fairly evenly and where the deposit has been almost completely excavated. In cases like that, MNI tells us something useful, such as how many animals were killed for the feast, or how many people could it have fed. In cases like that, the minimum number of individuals is probably pretty close to the actual number of individuals represented in the deposit. The most serious problems with MNI result when we try to use it not to estimate how many animals contributed to a deposit, but to estimate the proportions of taxa in an assemblage at a site. In such cases, we would overestimate the importance of rare taxa because a taxon represented by 1 or 2 bones can have an MNI no lower than 1. Meanwhile, we'd be ignoring most of the bones and bone fragments that come from the most abundant taxa, because they don't contribute to the MNI value. To complicate things further, MNI varies with the scale of aggregation. Your total MNI will vary depending on whether you calculate it by individual excavation unit, by small groups of units, or by whole sites or layers. In this example, calculating by individual excavation unit might lead to an MNI of about 11. But if we were to aggregate adjacent units, we would probably get a lower value, such as 8. If we were to aggregate into only 2 spaces, it would be lower still. And if we were to aggregate at the level of the entire site, it would be yet lower. The same thing happens if we aggregate stratigraphically. In this case, it usually makes sense to calculate MNI separately for each stratigraphic layer. But if, for some reason, we calculated MNI for all layers taken together, we would get a lower value of MNI than the sum of the MNIs for the individual layers. Much as these problems affect the values of MNI for individual taxa, they are compounded when we convert those values into percentages or proportions. Two other methods of quantification that are applicable to pottery involve attempts to talk in terms of whole vessels rather than just sherds. One of those is minimum number of vessels, which, as I mentioned earlier, is very similar to minimum number of individuals in faunal collections. And the other one is estimated vessel equivalence, which is a newer method. Both of them, however, have an element that involves measuring the diameter and preservation percentage of a whole circumference of rim sherds. You can actually use this with base sherds as well, but it's more normal to use it with rim sherds, in part because rim sherds tend to be more common in archaeological collections. So if we were going to quantify the same types of vessels, or same types of pottery that we saw earlier for NISP, we have to remove all the sherds that are not rim sherds. So now we're only looking at rim sherds, and each in turn we would stance on a diameter chart like this one and determine either its interior or its exterior diameter. So here I'll use the exterior diameter, which in this case happens to be 5.5 centimeters, and the preservation is 17.5%. So it's 17.5% of a full circle. So I'd record that on a laptop or something over here. That particular arc is at 19 centimeters, so we would estimate the interior diameter of this particular potsherd as being 19 centimeters, and the preservation of it is pretty close to 20%. This nice painted rim sherd, we do the same thing. Again, we have to decide whether we have to be consistent. We said we were going to use exterior diameter, so I have to stance it on here, and it looks like it matches up with 8 centimeters, and it has 10% preservation, and so on. We do this for all the sherds, and then we accumulate that information in terms of, we keep track of the diameters, and then for each group of diameters, in the case of MNV, we would calculate the total, the sum of the circumferences for each small range of diameter. For example, we might have 18 rim sherds at a diameter very close to 25 centimeters. In this case, their preservation sums to 128%, and we would round this up to 200%, or an MNV of 2. We would do this separately for sherds with different diameters, then round up the values for each diameter group, so that, in this case, we'd get an MNV of 3. Much as with MNI of animal remains, MNV works better on catastrophic deposits, such as cases where a settlement was destroyed in a conflagration, or the roof fell in on a building that was filled with vessels. MNV can work well where we have this so-called Pompeii effect, and there aren't too many sherds missing from our collection. When you have a catastrophic deposit, like the one you just saw, where it's likely that a large number of vessels were left in place when perhaps the roof caved in on a building or something like that, the minimum number of vessels is probably fairly close to the actual number of vessels that were in the building when the thing collapsed. So here, for example, we have just some of the rim sherds from that deposit, and more of them are on the shelves over here. And you can see that we have, in many cases, most of the entire circumference for some of these rims. In fact, probably almost all of them. We would have the entire circumference once we get them all fitted back together. But even if we didn't fit them all back together, we actually are working on that. But you can see that these things are substantially complete. So we would expect that even though there might be a few rim sherds missing because they lie just outside our excavation area or something like that, most of the rim sherds are going to be here. So if we were to total up the circumferences for all the rims of a particular diameter and then round up as you do with minimum number of vessels, we would probably get something pretty close to the actual number of vessels that contributed to the deposit. Estimated vessel equivalents can be a good measure to use when you're not interested in estimating how many pots there were in a particular deposit, but rather estimating the proportions of different vessel types to one another. So here, for example, we have 4 categories of pot sherds, or pottery types. One that has decoration on it, the other ones do not. These ones are wheel-made, these ones are wheel-made, this one is handmade. And we're not trying to identify individual vessels in this case, so some of these sherds might come from the same vessel and maybe not. It doesn't really matter for our purposes because we only want to know the ratios between the vessel types. So that one looks like it's about 15 centimeters and just shy of 10%. So let's say about 9.5% preservation. So we would jot that number down. And so that one, since there's only one sherd, it would be an EVE of 0.095 or 9.5%. Let's say keep it in percentages. For this type, we have several sherds, so we'd have to sum their percentages. So let's see, this one's kind of oval-shaped and fit there, so that's about 7.5%. And you will notice that these vary slightly in diameter, but we don't have to separate them by diameter in this case because we're not trying to figure out how many vessels there were, only vessel types. So if they vary a bit in diameter, it doesn't really matter. So this one, for example, is much smaller diameter. It's only about 12 centimeter diameter, and it has a 15% preservation. Now the reason we have to do the diameter is because the percent of preservation will vary depending on where you place the sherd on the diameter chart, right? So you have to match it up to the correct diameter in order to get the correct figure. And then this one looks like it's about 18 centimeter diameter, and it's about 16%. So we would sum all those up, which might come to a little bit over 1, I think. I didn't add them, actually. And we do the same for the other vessel types. So we now have figures for each of these, and to do the proportions of the different vessel types, once we have summed the EVEs for each of the types, we then divide that EVE by the total EVE for all types. So it's a very simple measure to do, and it's also an unbiased measure of the abundance of the different vessel types in the assemblage. Going back to the same example we used for MNV, with 18 rims, and in this case we don't have to care about the diameter, we would just use the 128% as our EVE value, or 1.28. Again, because diameter doesn't matter, in this case we can just sum all of the individual EVE values to get an EVE for the type. Once we've summed the EVE values for each type, We then divide by the total EVE for all types to obtain unbiased estimates of the proportions or percentages of each type by vessel rather than by sherd. Some archaeologists have adapted estimated vessel equivalents to other kinds of material culture that get broken. For example, a complete retouch tool would get an estimated tool equivalent of 1. The distal end of a tool would get an estimated tool equivalent of 1/3, as would a proximal end. A sample that had both the proximal and distal ends of a tool but was missing the medial portion would get an ETE of 0.67, as would a tool that was 2/3 complete. Even the more sophisticated version of MNI, even though it gives a somewhat higher value, probably grossly underestimates the actual number of animals that contributed to a faunal assemblage or the actual number of pots that contributed to a pottery assemblage, because it's a minimum value. It's the bottom of a range, and in most cases it's quite a bit below the actual number that contributed to a sample, unless the sample fraction is very large, or it's something like a catastrophic deposit and that has been virtually completely excavated, so we have almost all the bones from almost all the animals that originally contributed to the assemblage or to the deposit. In most typical archaeological samples, we have much smaller sampling fractions, and the probability that we have even one bone from all of the animals that contributed to that deposit is fairly low. So some archaeologists have taken seriously the fact that we sometimes have matching pairs of elements probably from the same animal, in order to come up with a measure that does actually estimate the actual number of individuals that contributed to the deposit, rather than just providing a minimum value. Two of those measures are called the Krantz estimator and the Peterson index, or Peterson estimator, and they both depend on this idea of matching lefts and rights in the case of animals. And basically they view the assemblage that we see in front of us as the result of a sampling process. From the statistical perspective, the probability of the left element of any bone in an animal entering the assemblage is the same as the probability of the matching right element being in that assemblage. The basic assumption is that if these bones are a random sample of the bones in the site, that lefts and rights have an equal probability of being included in the sample. And we also know that every left must originally have had a matching right, and every right must originally have had a matching left. And we can say that, on average, all the lefts have an equal chance of being included in the sample, all the rights have an equal chance of being included in the sample, and all the lefts have just as great a chance of being matched with a right as the rights have of being matched with the lefts. And we can look at this from a probability perspective and use that in order to come up with an estimate of the actual number of animals that must have been sampled to create the assemblage that's before us. To summarize, when the numbers of lefts and rights of the same element are unequal, that proves that elements are missing. If there are half as many rights as lefts, that would imply that at least half the rights are missing. But in that case, why wouldn't half the lefts be missing as well, since they have equal probability of being included in the sample? Furthermore, with such high probabilities of missing things, we would expect that some animals might not be included in the sample at all. In the original population, we know that the number of left elements should be equal to the number of right elements and the number of paired elements, because in living animals, there would be a matching right for every left. But in a typical sample, we'd expect some animals to be only represented by the left element, some by the right element, and some by both the left and the right. Meanwhile, we'd also expect some animals not to be represented in the sample at all. If we assume that the presence of lefts and rights in the sample are independent, the probability that any element will not be in the sample should be the same as the probability that it'll be missing its match. To look at it another way, that means the number of lefts expressed as a fraction of the whole population is the same as the number of pairs expressed as a fraction of the number of lefts. If we rearrange the terms of that equation, we get n equals L squared over p, and since we assume that right elements have the same probability as left elements, we can conclude that n is also equal to r squared over p. The Krantz estimator just averages the values for the left and right elements, so that we get L squared plus R squared divided by twice the number of pairs. The Peterson estimator uses much the same logic to come up with a simpler measure: the number of lefts times the number of rights divided by the number of pairs. This gives lower estimates than the Krantz estimator, except when L and R are equal. In this example, with 4 taxa, we get Krantz estimations of 68, 87, and 33 for the first 3 taxa, but we're unable to estimate the 4th one because there are no pairs. Using exactly the same data for the Peterson estimator, we get values of 54, 66, and 32. As you can see here, the Peterson estimator gives lower values than the Krantz estimator does, but both give much higher values than MNI would have done had tibiae been the most abundant element. While both the Krantz and Peterson estimators have the advantage that they estimate an actual parameter of interest, the number of individuals that contributed to an assemblage, they do also present some thorny problems. Not only are they only applicable to cases where things occur in pairs, we have to assume that lefts and rights are well mixed and come from random samples. When we match pairs, we can't be absolutely certain that the matches are correct, unless they're actually attached to one another, as in the case of the left and right sides of a mandible. But when they are attached, that violates the assumption that the assemblage is well mixed and comes from a random sample. Meanwhile, when the number of pairs is low, the standard error is very large, and when there are no pairs at all, we can't calculate either the Krantz or the Peterson estimator. Finally, it's not at all clear what we should do when different elements yield different estimates. Ubiquity is just the proportion of sample elements or sediment volumes that have more than 0 of the item of interest. In the example shown here, 3 of the beakers have no seeds at all, but 12 out of 15 beakers do have at least 1 seed in them, yielding an ubiquity of 80%. Ubiquity is a measure that's most often used by archaeobotanists, who often use a standard volume, such as 10 liters of sediment, for flotation. As I noted in my videos on sampling and probability, seeds with very different densities could have the same ubiquity values, and most importantly, the ubiquities would be very different if you used a different size of sampling element, say 1 liter instead of 10 liters. The key thing to remember is that ubiquity doesn't measure the abundance or even the importance of different taxa, but rather whether each taxon is spread out across a site or a set of features, or tends to be concentrated in a small number of spaces or features. Diversity is a measure that archaeologists have borrowed from ecologists, who use it to characterize things like forests and animal populations. Not surprisingly, we often use it to characterize archaeological faunal remains, but it can also be used for other things, including assemblages of pottery or stone tools. For example, when we want to know whether an industry is specialized or not. Diversity effectively has 2 components. Richness is just the number of taxa or categories in the population, while evenness has to do with whether or not those taxa are relatively equally represented, as in this example, or the population is dominated by only 1 or 2 very abundant taxa, as in this example. In extreme cases, there may only be 2 or 3 taxa. There are several different measures of diversity, each of which has disadvantages, and often a graphical approach is most effective. Here, in an example comparing 2 populations, when we plot the cumulative proportions of each taxon against the number of taxa, or richness, population A is clearly more diverse, even though it has fewer taxa than population B. Among the things I'd like you to take away from this video are that quantifying archaeological artifacts and other remains is tricky, in part because we only have samples of broken and otherwise poorly preserved things that were once part of pots or tools or animals. It's not difficult to count these things. What's difficult is determining what those counts might mean in terms that are really of interest. Often, we're not really interested in how many bits of rim or fragments of deer bone are scattered around a site. Instead, we might be interested in the percentage of a household's pots that were cooking pots, or the importance of venison in that household's diet. Unfortunately, those things are several steps removed from the fragments that we actually find in a site. Just counting fragments in a sample might be useful for some kinds of comparisons, but might not be adequate if we really want to know about the proportions of whole pots or about the dietary contributions in terms of energy or mass of meat. Some measures are perfectly fine for characterizing samples of fragments, but have low external validity, which means they're not as helpful as we'd like for drawing conclusions about things we could not observe directly. Some, like minimum numbers, are fine for describing particular cases where the sample is relatively complete and relates to a specific and often catastrophic event. But they're biased if we use them to estimate proportions of different taxa, especially when sample sizes are small. Others, like the Peterson index, are theoretically better for estimating relative abundances in populations, but they have serious practical problems, like the need to have matchable pairs, the difficulty of matching with any certainty, and the paradox of obtaining different results for different skeletal elements. Probably the best of these measures, when the goal is to estimate the proportions of whole pots, tools, or animals in a population, are ones like EVE that control for preservation in a mostly unbiased way. However, the selection of an appropriate measure for quantification will always depend on the goals of your particular research project.